This might be the one that goes viral.
What do you mean this one, Nick? The other ones are huge.
They are very big.
More than 100's viral, right?
Yeah. This might be the tipping point.
I hope we don't crash the website. Could crash YouTube.
Definitely.
The other ones are interesting because one of them I went back and I looked at it and there was like 100-something views. And I went, either other people are looking at it or the same students looking at it like 5 times. Either way, it counts, right? Yeah, good. Yeah. I'll make sure I note this on my Facebook page.
Share it.
Not that I have Facebook, but if I had Facebook, I'd put it on there. So here's the outline. This is what we're going to do. We're going to review lipoprotein metabolism. Then we're going to talk about a process called ApoB mRNA editing. And this is something that we have not discussed before. So I'll talk about the mechanisms of that and then relate it to cardiovascular disease. Then what we're going to do is after today, you'll have your lipoprotein values. And so we can compare optimal lipoprotein values to typical values, and we'll see it's not always the same. Then we'll talk about a little bit about LDL cholesterol and atherosclerosis, and then lipid-lowering medications. We'll talk about the mechanisms of how they work and what are some of the potential complications from those. We'll look at potentially your favorite slide from 328, and I'll go in some more details on some of this and less detail on other parts of it. So we know over here that when you consume dietary lipids, they are incorporated into chylomicrons and enterocytes, and the chylomicron then goes into the lymph and then in the blood. It then goes through the blood in capillaries and tissues, and that's where it interacts with lipoprotein lipase, and that enzyme hydrolyzes the triglycerides into fatty acids and glycerol. So the fatty acids can go into cells in mammary tissue, muscle tissue, and adipose tissue. It's mostly going to end up in adipose tissue. That form of lipoprotein lipase has a higher Km than the others, so it'll tend to end up in adipose tissue. Okay, so when you eat dietary lipids, that's how they're basically absorbed and then transported. In order to do that, chylomicrons need apolipoproteins. The main apolipoprotein in these guys is ApoB48. They also have what other one? Which other apolipoprotein? C2. C2 is the one that activates lipoprotein lipase. So they have ApoB48 and ApoC2. Now once chylomicrons hit the bloodstream, they're cleared typically within a few minutes. It takes a few hours for them to be made in enterocytes, go into the lymph, and then the blood, but they're metabolized really quickly. Now you guys fasted before you gave a blood sample, and one of the main reasons to let the chylomicrons clear. So when we talk about your blood cholesterol that we measured, we're not talking about the cholesterol that you just consumed. We're talking about the cholesterol that's been in there for, pre-fasting. We're also going to measure your triglycerides today. Well, that's what we're going to measure today. Okay, so that's one of the reasons we fast, is to let the chylomicrons clear. Chylomicron remnants are then taken up by the liver, and that's how the dietary cholesterol gets to the liver. The next lipoprotein up here is VLDL, very low density lipoproteins. Remember, they're also triglyceride rich. That triglyceride, most of it comes from the liver. Remember which apolipoproteins they have? ApoB100, and they need C2 as well. Good, because they're going to go into the blood, interact with lipoprotein lipase. Same thing, triglycerides are hydrolyzed, dropped off in these tissues. Then we have what's called a VLDL remnant, aka IDL. That VLDL remnant has two options. One is cleared by the liver, basically absorbed, metabolized by the liver. And what did we say here? Part of them can become LDL. Remember that percentage? About half. About half become LDL.
Do you want us to remember those percentages?
No. It's a fun fact. Cliff Clavin fact. You guys even know who that is? Wow. From Cheers? Yeah, there you go. Yeah. Yeah. You guys know who Seinfeld is?
Yeah.
All right, that's a little bit. That's yeah. Friends.
Oh yeah.
What other big series?
Maybe the next couple of years you won't have that.
Yeah, I know. I'll have to keep moving on. Now, those LDL, they can either go to the liver. This one you do want to know. About 75% of them are cleared by the liver. That's a really important part of their metabolism. About 25% of them end up in other tissues. Remember how they're taken up by cells? LDL?
Yeah, through the endocytosis.
Yeah, receptor-mediated endocytosis. And we talked about that, and we said as the cell's cholesterol goes up, they'll tend to make less LDL receptors. So I want you to think about, I'm going to put this in your mind right now and then we'll talk about it later. If we can control the amount of cholesterol in liver cells, that can have a big impact of the amount of LDL that stays in the blood. If we have a way of decreasing hepatic cholesterol, we can significantly affect the amount of LDL that are taken up by the liver. There's a few ways we can do that. We'll come back to that. The last one is, what's the apolipoprotein here, LDL? Just B100. Now we have HDL. HDL is the one we say they're involved in reverse cholesterol transport because they're taking cholesterol from extrahepatic tissue, taking it to the liver for the likely excretion. So they're kind of taking extra cholesterol from cells to the liver for excretion. So in general, we say, well, it's better if you have high HDL cholesterol. Because if you measure that, that's the cholesterol that's on its way to the liver. You measured that last week. Today, you're going to calculate your LDL cholesterol. In general, we say low LDL cholesterol is good because that could be potentially atherogenic. Now, VLDL are metabolized within a few hours. Once they become LDL, though, they can stay in your blood for a few days. HDL somewhere in between, a few hours and a few days. Okay, now, what's next? Why are humans so susceptible to heart disease? Number one lethal disease in the U.S. and most of the Western world is heart disease. Some nice things about being human. This is not one of them. But we're going to talk about this process. One major factor that has to do with heart disease is apolipoprotein B. In humans, chylomicrons contain ApoB48, and LDL contain ApoB100. The form of ApoB significantly impacts lipoprotein metabolism. Lipoproteins that contain ApoB48 are cleared faster. They tend to be taken out of the blood faster. I'll explain the relevance of that in a minute. A really interesting part of this is B48 and B100, two different proteins, are made from the same gene. One gene, two proteins. So how are they both made from one gene and how does it relate to heart disease in humans? That's what we're going to go through. Yeah, this morning, this was not showing up right. Now the text is correct. It's just a weird thing. I don't know. It should be, yeah, it's eerie. All right, so this is called ApoB mRNA editing, the process we're going to talk about. ApoB mRNA editing occurs in human small intestine, but not the liver. And as the name implies, what happens is the ApoB mRNA is made, and sometimes its sequence is changed. So I'll give you the details of this. So here's the ApoB gene. One of the largest genes in the genome. ApoB is a massive protein. Huge gene. So here's 5' end of the DNA, 3' end over here. Here's about the central part of the gene. There's a codon, a CAA codon. Do you know what that encodes for? God, that'd be impressive if you did. Not likely. I can barely remember this myself. That's a glutamine. TAA, what's that? That's a stop codon, because when that's converted to RNA, it's a UAA. Remember that? No? Okay, that is. UAA is a stop codon. So let's look what happens. In transcription, we go from the gene, we go to an RNA, and then an mRNA. And all this happens in the nucleus. With no editing, in other words, native transcript, native sequence, that CAA remains. So now that mRNA goes into cytosol, is translated into this protein. So when the ribosome is moving through the mRNA, or vice versa, when we get to this central part, ribosome puts in a glutamine, keeps going, until we get to this stop codon. And that's it. Poly A tail, we're done. This is ApoB 100. This is what happens in human liver. In the intestine, how do we get B48 and chylomicrons? ApoB gene is transcribed, it's edited. So in the liver, the C is changed to U. Sorry, in the nucleus. Sorry, in the nucleus. Glad you guys are paying attention. In the nucleus, that cytidine is converted to uridine. That changes the codon from a glutamine codon to a stop codon. So it's edited in the nucleus. In this case, the nucleus of intestinal cells. Then the mRNA goes to the cytosol, is translated. When the ribosome's moving 5' to 3' comes to this, what happens? Translation stops. It's approximately the halfway point. So you end up with a protein that is 48% the size of B100. That's why it's called that.
Where does it change from nucleus to cytosol again?
Post-editing.
Okay.
Yeah, so translation, cytosol. So editing is post-transcriptional, pre-translational, right? Post-transcriptional, pre-translational. Here's another way of looking at it. Little different detail. This is ApoB mRNA editing. It involves a cytidine to uridine conversion performed by a cytidine deaminase. This is a category of enzymes, and as their name implies, they deaminate cytidines. If you remove an amine group from cytidine, what do you think you get? Uridine, uracil, right? So the enzyme just takes an amine group off. That's what converts it cytidine to uridine. The enzyme that does that is ApoB1. ApoB1 stands for ApoB. So this will be apolipoprotein B editing, catalytic, subunit or component, 1. ApoB, oops, sorry. mRNA, editing, catalytic subunit 1. There's another way of looking at it. There's that codon that's changed. Now, again, this is in humans, in the liver.
So is that DNA is only located in intestine then? Like it just doesn't exist in the liver at all?
In humans, that's correct. Yeah. So good point. We'll get to that in 1 second. Here's the liver. It's unedited, so we end up with ApoB 100. So which lipoprotein has this? VLDL. And LDL, because it comes from VLDL. Over here. In the intestinal cells, the unedited mRNA is edited. CAA converts to UAA, it's a stop codon. So you end up with ApoB 48, so it has 2,152 amino acids. Still a big protein, but about half the size of ApoB 100. ApoB 100 is one of the largest proteins in our body. There's only a handful of proteins that are bigger. Okay? Now, I remember when this first paper came out, 2 papers came out simultaneously. Labs were sequencing the ApoB 100 gene, and then at that point we were just able to start doing sequencing from mRNAs. What was interesting is they came up with sequence alterations between the gene and the mRNA. They repeated it several times, and I know this because we were collaborating with one of the labs. When I was at University of Arizona as a grad student and then a researcher, we were collaborating with a lab in Baylor. It was one of the top lipid metabolism labs in the country. So when these papers came out, they realized initially they thought it was a mistake, and then they realized this is the first evidence of anything like this occurring in mammals. The only other incidence was in fungi. They found some editing. But what this means is this is a controlled event, and it affects our risk for cardiovascular disease. So what's driving this is, one of the things I said was, lipoproteins that contain ApoB 48 are cleared faster. When you start looking across species, for example, we obviously have heart disease. Rats don't. The reason rats don't develop atherosclerosis is because they edit in their liver. About half of the VLDL that rats make have ApoB 48. About half have B100. So you can do these assays, and I used to do these in grad school. You take the mRNA, you isolate it, and when I would do this in rats, I set up what are called ApoB mRNA editing assays, and you can quantify the amount of this cytidine to uridine change. And in rats, you usually end up with approximately 50 to 60% editing in the liver. They figured out the enzyme that does that, ApoB1, is basically what controls this. The bottom bullet point says this addresses Alex's point. Tissue-specific expression of ApoB1 determines whether or not editing will occur. If ApoB1 is in a tissue, ApoB mRNA gets edited. Do rats express ApoB1 in the liver? Yeah. Do rats express ApoB1 in their intestinal cells? What do we make in our intestinal cells? Chylomicrons. Which apolite protein do they have? B48. Rat intestinal cells express ApoB1. They put ApoB48 into their chylomicrons. In humans, do humans express ApoB1 in the liver? No, because we don't edit. All of our VLDL have ApoB48. What about our intestinal cells? Do they express ApoB1? Yep. Because we make chylomicrons with P48. What if we could edit in the liver? What if humans could express ApoB1 in the liver, make VLDL that have B48? Could that significantly increase our protection against cardiovascular disease? Absolutely. That was a huge research topic, and this is years ago now, because the theory was if they can use gene therapy, you guys know what gene therapy is. Gene therapy is sometimes when you can incorporate a normal gene into a tissue and express it to compensate for abnormal genes or mutations. A classic example, PKU. When I was doing research before I came here at OHSU in Portland, the lab next to ours was working on PKU. So they were using viruses to contain the normal copy of phenylalanine hydroxylase and then infecting the mice with those viruses so that they would express increased levels of phenylalanine hydroxylase in the liver. And it was working pretty well in mice, and I haven't heard followed up on that since then. So the theory was, in humans, we could do the same thing with ApoB1. We could take a virus, disarm the virus, put in the ApoB1 sequence, infect people, cause their liver to express ApoB1 and make VLDL with ApoB48. The push was huge because it could have significantly decreased cardiovascular disease risk, could have saved millions of people cardiovascular disease in theory. Here's the problem. Go ahead.
Would that mainly be aimed at those individuals who have genetic issues with cholesterol?
When they first did this, and this is kind of scary, and I don't know all the details, but I got word of this, and I actually read a case study somewhere. They had two individuals. They had really, really high cholesterol values, and they were considered to be acceptable candidates to try this therapy. I don't know if this happened in people, but about the same time, they figured out in mice they were increasing the amount of ApoB1 that was expressed in tissues, but by doing so, they were increasing the risk of cancer. The mice had increased tumorigenesis. And here's the theory why. Here's ApoB1. This is called the minimal editosome in vitro. Editosome means what's required in order to edit ApoB mRNA, and this is in a test tube. So basically, if you want to do gene therapy, you got to figure out how this thing works. Here's ApoB1. See the yin and the yang? ApoB1 functions as a homodimer. Okay, there's two copies. They bind to each other. Then they bind this protein right here. See this big blue part? This is a complementary factor, sometimes referred to as either P66 because of its size or auxiliary complementary factor. We can just call it complementary factor. The complementary factor provides tissue specificity for ApoB1. So ApoB1 is a cytidine deaminase. It doesn't recognize where it's supposed to edit. Complementary factor does. So complementary factor looks for this nucleotide sequence. It then locates ApoB1 at the appropriate place. This sequence is called a Maureen sequence. It's based on one of the sequences from fungi, I think. This is conserved. So for example, we did studies, we published this in Journal of Nutrition a long time ago. We were working with hamsters because hamsters have a similar lipoprotein profile to us. It's closer than rats. So what we did is we cloned ApoB1, determined its sequence, we cloned this portion of ApoB mRNA, and then compared across species. We found ApoB1 sequences were pretty similar between species. These were a little bit different depending on whether or not that animal actually edited. It kind of depended, but overall, pretty highly conserved through here. Now, if you look at what's happening, it's important that this sequence is right for the complementary factor to recognize, because look, right here, there's another C. If you change this to uridine, what would happen? You could change this codon and change the protein. So it's extremely important that ApoB1 edits the correct C. What they think was happening was when they were overexpressing ApoB1, it lost its tissue specificity. It was making abnormal proteins because not only was this being edited, so were other parts of ApoB, and so were other transcripts. So they were editing proteins that shouldn't be edited.
Would that be because it's like saturating that area, like it doesn't have enough
Complementary factor.
factors?
Yeah.
So, but in humans, if you found like a particular like low dose number, can you reduce the risk of cardiovascular disease if you're not over adding the ApoB?
In theory, I don't know why, but all of a sudden they left this area of research. I think because they felt like this is not something that is going to be feasible for whatever reason. And so these are multi-million-dollar labs that were chasing this, that had huge grants, and everybody pushed it to this point, and then we got to the point where some of that increased risk of cancer came out and everybody left. So apparently some of the people just determined it's not worth going there.
A lot of research labs, they don't want that label either, right?
Yeah, and yeah. And what's interesting, we were collaborating with this lab that was huge, and they had people in there that were phenomenal researchers. The person that first cloned this, her name is Bobby Tang. She's from Taiwan, man. She was an incredible scientist. They had another person that figured out this ApoB1 bound together. He was actually a guy from Hong Kong, and they had all this research going into this one area, and they were extremely well-known lab. It was the Chan lab, Lawrence Chan, and everybody wanted to go there and work there. And then all of a sudden, all these people just left because of that. And Lawrence Chan moved into other areas of lipoprotein research. He dropped this completely. And I think he just kind of realized, I'm not going to get any more money for this. It's not worth it, and not something that we're going to do anymore. So it was all of a sudden everybody left. We were characterizing at that point. What I did is I wrote grants to look at how zinc levels affected it, because ApoB1 is a zinc binding protein. So that's one of the reasons we stayed in it for a little while after that, to see how zinc might affect that. And actually my PhD research was on copper status and how it affects ApoB1 levels and editing. And then I did some stuff in hamsters where we looked at zinc levels in that as well. Hamsters are interesting to work with, and originally I thought, I don't want to work with hamsters. Man, the people pets and everything. That's not very nice. And then one bit me, and I went, all right, I'm okay with this. So it's one of those, and I mentioned this, but rats don't get cardiovascular disease, hamsters do. They develop atherosclerotic plaques. So the model that you use to study this is really important. So that's kind of what happens in people, is because we don't express ApoB1 in the liver, we make VLDL with ApoB 100, and that sets us up for cardiovascular disease, because our LDLs stay in the blood for longer periods of time and they can be modified. And because of that, that increases the likelihood that macrophages are going to target them and use them for, and basically causes lipid accumulation in the, sometimes in the tissues, in the arterial intima. I think we're good there. Basic overview, remember this one? Chylomicrons, VLDL are the triglyceride-rich ones. LDL and HDL are the cholesterol-rich. Now, we looked at this before, but now let's put it in the context of your values. This distribution of triglycerides and cholesterol among fasting plasma lipoproteins. So in other words, in this column, we're looking at cholesterol mg per deciliter, same units you guys have. This is plasma instead of serum. Basically, it'll be the same. This is kind of a typical value that you see in reports. So this is our total cholesterol, so we set that at 100%. So one of the questions is, why are we doing the different fractions, and what should we expect as far as cholesterol values in each of the fractions? Well, here's VLDL. Of this total, only about 5% of it is in VLDL. Why?
It's being converted.
It's fasted. These are fasting values. They've been metabolized. Now many of them are left in the blood. Some of them have been converted to IDL, but IDL is even less. It's about 2%. Why? Metabolized quickly. LDL, look at this. 115 mg per deciliter. Kind of a typical value. 65% is kind of a typical value in most humans. We carry most of our cholesterol in LDL, and that's the particle that can hurt us. So we're what are called LDL animals. Rats are HDL animals. Here's our HDL. Kind of a typical value, 45. That's about 25% of our cholesterol is in HDL. What I want you guys to think about today is when you get your values, look at this. If we add up LDL and HDL, what percentage should it be total? 90%. Look at your values today after you calculate your LDL cholesterol. When you add LDL and HDL, it should be about 90% of your total. That's a way that we can kind of say the assays are probably working correctly. So you'll have that in your actual report. Now, I'd say in general in the past, 329 labs have had a total cholesterol of approximately 175 to 180. And what's interesting is, I'd say because of the demographics in here, HDL tends to be higher than average. Women tend to have higher HDL than men. Why? Other than, you know, you're nicer. Why?
Does it have to do anything with their child?
Estrogen. Estrogen tends to increase HDL cholesterol. So what happens post-menopause? In women, lipoprotein levels can really change post-menopause.
Do they become lower than men's at that point? Because it's not.
Depends. They can, yeah, they can. What tends to happen though is your total goes up, so LDL tends to go up, and that kind of changes that ratio. So then you kind of become even higher in your LDL to HDL ratio than men in general. So it kind of reverses. Men, we tend to die a slow chronic death, right? So our lipids are not as good as yours through most of our lives, and then that's one of the theories behind why women live a few years longer is that HDL protective effect. That's one of them, among others.
Is estrogen therapy ever used in men to kind of help finances in real severe cases?
I haven't seen that. No, I haven't seen that. I think, you know, one of the issues with those hormones is they need appropriate receptors. And if they don't, and those hormones start binding other receptors that they're not really the natural targets for, that can cause problems. So I think you're really careful on stuff like that. Now another thing that's interesting is some of you guys will have HDL that's higher than LDL. There's a term for that the cardiovascular people use. I can't remember that off the top of my head. That's kind of rare, but some of you might actually have that. In general, it's LDL that's higher even in women, but some of you might be close. And I've even seen in our research studies when I'm watching the women go through, you can see some of those women definitely have higher HDL than LDL. So, that's all good. Typical values are what we kind of use as values and categories is shown here, and you guys have this in your handout as well. Blood cholesterol levels. Oh, let me mention this, sorry. Triglycerides. You're going to measure triglycerides today. They have 85 mg per deciliter up here. We're saying that's 100%. Where are they? Well, remember VLDL is the triglyceride rich. So if there's any VLDL there, they're loaded with triglycerides. So that's about where half of the triglycerides are in the blood. A little bit in IDL, a little bit in LDL and HDL. Remember, they're mostly cholesterol. Today's values, make sure you do it right. You're going to get, some of you are going to get absorbances that are pretty close to the standard. Some of you are going to get really low absorbances. So when you put in your serum into that tube, it might not turn pink very much at all. So make sure when you put that in there, mix it, move your tube in the rack so you know you did that one. Last week when you're doing the cholesterol, turned pink fairly quickly. These might not. So your absorbances today might be as low as 0.040 or 0.030, and the standard is going to be like almost 0.3. So there'll be quite a bit of a difference. So make sure your cuvette's in there right. Make sure you did the assay right. There's going to be a wide range on this. Some of you today might be quite a bit higher than 85. Don't worry about it. We're going to look at all of this kind of together. Some of your values might be a lot lower than that. So triglycerides tend to be highly variable in this stuff. In general, though, we say we want it to be below a certain level, which I have in your handouts. Now, for these desirable levels, we say less than 200 total, less than LDL, less than 100 for LDL, and greater than 60 for HDL. In general, higher the better for HDL, lower the better for LDL. Borderline risk, total between 200 and 239, LDL between 130 and 159, HDL 40 to 60. Elevated risk, greater than 240. Is that common? Yeah, it is relatively common. LDL greater than 160, HDL less than 40. 40 is kind of a cutoff for HDL. If it gets lower than that, then it elevates risk. I've seen them as low as, a friend of mine, his was 25 or 26, I think it was 26, and he was early 60s. And he showed me his values. He said, hey, you mind taking a look at these? And I met him through the gym and became friends with him. And he asked me one day if I would look at this, and I said, and I happen to know his diet was pretty bad. And he was overweight. And, I don't think he had ever had it done, though. So he brings it to the lab, I'm looking through them, and he's like, oh, so what do you think about these? And I said, oh, I wouldn't worry too much about it, blah, blah, blah. He goes, well, what does that mean? He said, that HDL is really low. I said, yeah. He said, have you seen him lower than that? I'm like, nope. What am I supposed to say? I said, no, I don't think I have. Started talking to him more about his diet. It was not very good. And one of the things we want to keep in mind, though, remember, this is multifactorial. There's other things that affect this. Lipids are part of the equation. You guys know your blood lipids now, or you should have a pretty good idea. You guys are kind of in a rare group of young individuals that know your blood lipids. Most don't. A lot of adults don't. Just like I said for blood glucose, I would encourage your family and friends to have a blood lipid panel done so they know that. So that way you're not like, they're not, or even your friends, not 60 years old and all of a sudden go, oh, my total cholesterol is really high. Okay? It's kind of the same thing for osteoporosis. We're going to do your bone scans. We're going to look at that. You guys will know your values. Some of your values will be low. You'll have lower than average bone density. Again, you're in your 20s or you're young in life so that you can actually cause an effect. I've done scans on people that are 70 years old, and at that point it's really hard. It's hard for them to deal with. So it's nice to know some of these things overall, especially at certain points in your life. So when we're looking at these values, keep that in mind. Other underlying risk factors for this: previous heart attack or diabetes tends to increase risk. 2 or more of the following are very high risk. Smoking, one of the worst things a person can do for their health. High blood pressure, low HDL cholesterol, family history of early heart disease. Age, for men over the age of 45, elevated risk. For women over the age of 55. Slightly different there. Remember, it's multifactorial. Are those typical values? So there's been lots of these studies done. I have results from one of them I'm going to give you real quick. This is the Minneapolis-St. Paul study. Any of you from there? All right. No one knows anybody in this study. 2,500 subjects for about 20 years. They followed these. So some of the findings were... $1980. 82, that's when they started. I I gotta tell you, the 80s were nice. 80s were a lot of fun.
Doing a lot of concerts.
Yeah, a lot of stuff going on in the 80s, right? So this is when they started. I don't know why it's important that I bulge that. What do you think their total cholesterol values were? Men. Be nice. Men. We know what desirable is.
190.
Thank you. 212. Women?
180.
Pretty close. Followed these individuals after they went to all the concerts, did all these things. Right? 20 years later, same cohort.
230.
Follows. Ruthless. 199. Went down. Women? Just kidding.
Oh.
Went down as well. Good for them, right?
The health crazy.
You know, the 80s and 90s, yeah, I mean, they're paying attention. Richard Simmons is doing his thing, right? All these health promotion campaigns and everything, and diet. Think that's what happened? No way.
What's the age of the individuals at this point?
Different age groups. They had different age groups. So
Oh.
overall, this is, yeah, overall average is this. When they started stratifying by age, they realized that not all the age groups went down over that period. The younger individuals didn't go down. Younger individuals, their lipids actually kind of went up. So when they started looking at other variables, a lot of people have hypothesized overall blood lipid values are probably decreasing because of increased prevalence of lipid medications. If you pay any attention to any kind of advertising, that is a huge market. They're extremely popular, right? Are they effective? They can be. We'll talk about that. But overall, that's what they said that's probably what's responsible for this. The reason? Because there are exceptions to that decrease in total cholesterol. For example, the age group of 25 to 34, they had elevated cholesterol. Why? One of the reasons is they probably didn't know their cholesterol before the study. They weren't on lipid-lowering medications, so theirs tended to be higher. Now, obviously for us, we want to see people try other things, right? We want a good diet, lifestyle changes, exercise, make sure they're doing everything that they can before they go on some type of lipid-lowering regimen of medications. So if we look at the conclusions there, blood cholesterol values are important to consider. It's part of the equation. They're important to try and control. But they should be evaluated with other aspects of health, like blood pressure, right? Exercise. We'll talk about insulin in a second. All of these other things that are controllable, we want to try to make sure that we control for them. Now, if a person has normal blood lipids, they can repeat the test every few years. Probably every 2 or 3 years is fine. If lipids are elevated, it's probably better to do it more often. Try some things and then wait a few months and then have it evaluated again. And I've known some people that have made changes. Like I remember my mom, she wouldn't mind if I tell you this. A few years ago, her blood lipids got really high. They went up over 300. Before that, they were usually around 200. It went up over 300, and fortunately, I think for her physician was determined to keep her off of meds. And she said, I don't want you on those. I think the side effects can be too harmful and nasty, so we're going to try other things. So she made some recommendations, and then a lot of people don't want to listen to things until they have to. So I noticed she started making some changes and kind of asked me about it. What do you think about this? So we made some changes, and it dropped significantly. So after a few months, she got back down to 200 again. So we'll talk about some of the diet stuff, but for the most part, what I hate to see is right away they'll go go on this medication. Like I remember a few years ago, it was probably 5 or 6 years ago, I went in with my dad. They had like a community thing, and so we had his blood pressure tested at the hospital. So I walked in there, and I was sitting there, and the guy goes, "You want me to do yours?" I said, "Sure." So it was high. It was like, I think he got like 142 over 100, something like that. I was kind of surprised. All right. He starts talking to me about antihypertensive drugs. You know, you really ought to think about blah, blah. I said, "No, I'm good." I said, "I normally don't test that high. You know, it's important that your regular stays on." I'm like, "No, I'm good." And finally I said, "Yeah, I think it's kind of concerning that you're talking to me about that so quickly, because this is the first test. It should be a repeated measures type of scenario, and I don't think it's really appropriate that we're talking about this right now. I hope you don't do that to everybody that comes in here, right?" Yeah. About a month later, I go into the dentist, and I'm sitting there, and they took my blood pressure again, and she goes, "Wow, it's really low. Is it always that low?" So you got to do this kind of stuff over and over again. And typically mine's pretty low. It's hard to believe, huh? If you go through a step on lipids, the same thing. So we want to do repeated measures where you're kind of consistently looking at it. And if you need to, do it every few years. Remember this? Response to injury? Uh-huh.
Why would they not have told the participants in the study not to go on the cholesterol-lowering medication? Because it's not an accurate determination of cholesterol levels over time.
Unless you're trying to figure out, you know, if that's an important variable. And I don't know why they didn't control for that. You know, sometimes you'll use that as an exclusion criteria. In that case, they probably had enough subjects to leave them in, and they can actually test for that to see if that might be one of the variables that's causing the change.
But it obviously was, right?
It seemed like it, but when they stratified for age, they found out, oh, okay, that's,
Not really the case.
yeah, that actually probably was the case, yeah, that it was,
Yeah.
yep. Yeah, so good point. Now, in those types of studies, that's kind of a longitudinal study where it's relatively easy to, as you think about it, when you start controlling for stuff like that, it's easier if the higher the numbers that you have on those kind of studies, the better. If it's an intervention, it's a little bit different. But in those types of studies, those large numbers can sometimes allow you to keep people in, and you can kind of control for it later. So if we look at this, response to injury theory, we talked about that in 328. Causes of injury include high blood lipids, smoking, hypertension. Remember, we have the injury, macrophage accumulate in the area. They tend to ingest modified LDL. That leads to fatty streak and plaque progression, and then eventually the plaque can rupture and either cause occlusion at that localized site, or that clot can travel and cause thrombosis in other regions. Overall, though, high plasma cholesterol can be a major factor in the development of atherosclerosis, so what can be done to keep LDL low? That doesn't mean if you have high LDL, it's going to lead to heart disease. It means it is one of the risk factors. A few things that we can do. Remember, the type of fat is consumed can affect plasma LDL. You guys are coming along at a great time where this argument is now heated up again. How important are saturated fats in the diet? There's quite a bit of data to indicate they probably are. For some people, saturated fat intake is more significant for their blood cholesterol than dietary cholesterol itself. So I think you definitely want to control for both. So in general, it tends to increase LDL. Remember the mechanism? LDL receptor. Saturated fats tend to decrease LDL receptors. Unsaturated fats, the opposite. So both monos and polys tend to increase LDL receptor numbers on cells. That draws it out of the blood. What about fiber? One of the things that we talked about before was that intracellular cholesterol in the liver kind of determines how many LDL receptors are on those cells. Fiber, when it's in the gut, if it binds bile acids or cholesterol, if it binds bile acids, remember it decreases bile acid recycling. That way more cholesterol is converted to bile acids than released into the blood.
Is that cholesterol just coming straight from HDL, or is it like the ideal cholesterol? Is it all pooled into one cholesterol in the liver, or how does that work?
The cholesterol can either come from the diet or hepatic synthesis, or some other cells can synthesize cholesterol as well. So it's kind of a mix. Remember, about 2/3 of the cholesterol in our body is from our own body's synthesis. Some of it comes from the diet. So that fiber, though, if it decreases bile acid recycling, that's one way it can decrease cholesterol in the liver. If cholesterol in the liver goes down, more of those LDL tend to end up in the liver. So that's one way to do it. Another way to do it is the lipid-lowering medications now can decrease hepatic cholesterol synthesis. By doing that, it increases LDL uptake. So they work kind of like fiber does in a way, but they're working through a different mechanism. I think I have what else? What else can we do? Maintaining a healthy body weight and exercise is also important. Being overweight or obese can lead to a reduction in insulin sensitivity or effectiveness. So that can lead to elevated insulin, at least for a certain time period. During that time, that insulin can actually increase hepatic cholesterol production. So insulin tends to increase cholesterol synthesis, and that can cause higher VLDL. In general, the more VLDL, the more LDL. Now, how it works. After binding to its receptor, insulin affects cholesterol synthesis by at least 2 mechanisms. 1, HMG, if we look over on the right side, remember HMG-CoA reductase? That's the enzyme that controls the rate-limiting step in cholesterol synthesis. Cholesterol is made from acetyl-CoA. Multi-steps and then converts to this molecule, beta-hydroxy-beta-methylglutaril-CoA, also known as HMG-CoA. So here's HMG-CoA. It's converted to mevalonate by HMG-CoA reductase. Insulin increases the activity of HMG-CoA reductase. Once you get mevalonate, a few more steps, you got cholesterol. So this is the point of regulation. So if insulin is chronically high, cholesterol synthesis tends to go up. And if this is in the liver and cholesterol synthesis is going up, that means those liver cells are going to make fewer LDL receptors. So less LDL is taken up by the liver. So LDL in the blood tends to stay higher. So insulin is actually increasing the expression of the HMG-CoA gene. That's one of the mechanisms. More expression. So it makes more enzyme and it activates more of it. Because insulin also activates the enzyme by dephosphorylating it. So expresses more enzyme and activates it.
Is there a link between diabetes and cholesterol?
Yeah.
Pretty strong correlation between the two.
In general, yeah. Tends to be elevated. Now, lipid-lowering medications. There's a class of drugs called the statins. The first statins were discovered in the 70s. There was a Japanese researcher that believed some organisms probably made molecules to inhibit the cholesterol synthesis of invading organisms. So they thought it was kind of a protective strategy of some organisms to downregulate cholesterol synthesis. So they start isolating these compounds, and then those initial studies were supported by a company called Merck. You ever heard of Merck? Merck is a huge pharmaceutical company now. It makes tons of reagents and medications. They continued to develop these, and they found a whole class of these molecules that are called the statins. So they're a class of medications with several different trade names, like Lipitor, Crestor, Lovastatin. There's about 10 of these really common ones on the market. The mechanism of action is competitive inhibition. Guess which enzyme they bind? HMG-CoA reductase. This is one of the statins. I think this is Lovastatin. I think I downloaded. Guess what this molecule looks kind of like, or pretty similar. Insulin's a hormone, but it's a peptide.
And that's,
Competitive inhibition. What would it look like?
probably HMG.
Yep, HMG-CoA. So the drug binds instead of HMG-CoA. So less HMG-CoA is bound by the enzyme, less cholesterol is made. Are they effective? You ever seen numbers on this or heard numbers on this? They will claim that anywhere between, in general, they'll see a reduction in LDL cholesterol of 40 to 75%. Sound like a lot? That's a lot. And that's probably accurate. So they can have a significant impact. And they are widely prescribed. Some of the controversy with these has to do with their side effects. Some of it has to do with the actual protective effects. The data indicates these are probably very valuable for individuals that already have atherosclerosis or have had a myocardial event. It's very protective against the second one. One of the reasons is not only do they decrease the amount of cholesterol in the blood, some of them can actually decrease the size of plaque.
How long do they stop working if they don't change their diet? Will it ever just like stop doing what it's supposed to do if the diet is still really bad?
No, because
It's like
kind of a
security
typical course of action is, here's your statins, here's your regimen, this is what you're going to be taking, and that's in combination with the diet that they consume. Yep. So a lot of times I would say probably not a dietary modification, right? So like I said, not ideal. For some people though, they probably should be on them. For a lot of people, don't need to be on them, shouldn't be on them. So should be kind of a last resort to me. Side effects, right? Kind of typical for drugs, right? They have side effects. Most common side effects for these. So the most common side effects for the statins are muscle pain and weakness. Can lead to protein breakdown in muscle.
Isn't that super bad as well for like the elderly, because they already worry about protein breakdown? It would just like further push that in.
You can, yeah. I think ideally, you'd want, if they have to be on this, you're going to titrate in a minimal amount that you can have the effects that you want, but not more than you actually need. So yeah, that would be one population. But I know people in their 30s or 40s that are on these. How long are they on them? Once you start them, you're kind of assumed to be on them for the rest of your life.
Especially starting that early.
Yeah. So now, and what I just said was, in the 70s and 80s are where these really started becoming popular. So what about long-term studies? Yeah, they're still kind of being done, right? So that's a side effect. Now, one of the reasons that can be so harmful is not only because of the pain, but if there's significant enough muscle damage, some of those proteins can get into the blood and lead to other tissues being affected, like the kidneys. So another side effect is kidney dysfunction. If that muscle breakdown is significant enough, it can lead to kidney failure.
It's not long, it's not length of usage of the statins, it's like a potential toxicity, like too high levels, or?
I'd say that's more related to not long term. Some of the initial things. And let me do the side effect first, and I'll come back to that. Another of the most common side effects is liver dysfunction. Right, I mean, these are not natural compounds in the body. There's no transporters for them, stuff like that. So they're lipophilic enough, they can go through cell membranes. So the theory is sometimes they will end up embed in membranes and actually affect the stability of that membrane. That's a theory behind the muscle damage. But remember, these can bind lots of other proteins other than HMG-CoA reductase. So ideally what happens is the individual has a baseline liver test done where they take a blood sample, look for the amount of hepatic enzymes in the blood, and then as they go through a drug regimen, they can monitor that to make sure that the liver's not being damaged. If it is, sometimes what they'll do is they'll switch them to another statin. Kind of scary, kind of weird, I should say. They'll switch to another one. Sometimes the side effects are less, so they'll try a few of these. So, how long would you be on these for you to see the initial changes? What's a minimum time frame? Yeah, happens pretty quick. LDL is only in your blood for a few days. When these drugs start going in and kind of equilibrating in tissue, the effects can happen pretty quick. So after a few weeks, you can see some changes in the lipids. But again, it's important that they're consistently monitored and everything. With all of that said, I will say that overall, per risk, elevated risk for drugs, these are not as bad as a lot of them. So overall, I think you could say their side effects tend to be relatively small compared to some other ones. Okay, so another reason why they become so popular. I think that's it. Any questions on this? Okay, so if you would, let's do this. We'll take about a 10-minute thing. That'll give Jamie and I a chance to get ahead of you guys. We're going to make standards and then blanks for you. Then she's going to put out a bunch of the reagents for you. So we'll do about 10 minutes. We'll start, and we'll do triglycerides. I'll read through that and get us all set up before we start. So we'll start about 1:30 on that clock.
So it works as it looks like the
My cholesterol is really low, but my HDL is really low. I don't know.
it's not the HMG.
Yeah,
So
because
that
we both
HMG
ended up with super
code
Yeah.
directly can't work.
and their reactions.
Yes, please.
Well, and I have a lipid panel done like a month ago, and it's about, I think the paneling on that was 52, and I got 35 from HDL on this one.
I think my total is low in that situation.
Yeah, so I don't know. But that's why he was telling us to like at the end add up your HDL and LDL, because if it doesn't add up to 90, then that probably means that.
I'm sure it's probably better now.
So we did
Yeah,
something
that-
wrong.
Diet and exercise. Yeah, no kidding.
That's what he said.
I mean, there are,
I
but
saw
they're not
like
healthy
people
and they're
walking
not a
around
good method to go about it. Yeah, it's not for like long term